Welcome to Roadmap to Residency, Evidence-Based Application. In this video, we will be discussing about drug-receptor interactions. So, whenever we talk about drug-receptor interaction, the first thing we need to know is the extracellular signaling. So, signaling molecules are released by signaling cells, and the signal is called the ligand. Any molecule that is bound by a protein, from the Latin liger, which means to bind. The ligand binds to a specific receptor on a target cell. This ligand receptor interaction induces a conformational or shape change in the receptor, and then it produces a specific response, which is called the cellular response. It can include a vast array of compounds, example, small amino acid derivatives, small peptides, or proteins. The ability of a protein to bind to or interact with a ligand depends on the formation of weak, non-covalent bonds between them. This process relies on the sequence of amino acid in each protein and the way in which their side chains or the R groups interact with each other. The different side chains can form different bonds. Because of this, the protein interactions can be very, very specific. Now, talking about basics of agonist and antagonist. So, the endogenous ligand bind to the receptor and produces its effect. The An agonist drug has an active site of similar shape to the endogenous ligand, so binds to the receptor and produces the same effect, that is called as agonist. Whereas antagonist is the opposite, that is, it inhibits or it blocks the effect. So an antagonist drug is close enough in shape to bind to the receptor, but not close enough to produce an effect. It also takes up receptor space and so prevents the endogenous ligand from binding. So now we know that whenever a drug binds to its receptor, it produces an effect. Now, agonist drug, they are similar towards that drug. Now they're similar towards that chemical, that endogenous ligand, and it produces similar effects. But antagonist is only similar in a way that it can bind to the receptor, but it does not produces the effect. The types of chemical interaction between the ligand and receptor. So there is number one, covalent interaction, that is formed when two different atoms share electrons in the outer atomic orbitals, and this is an irreversible interaction. And then there is a non-covalent interaction, that is an ionic bond, hydrogen bond, or van der Waals interaction. Now, one example is estrogen receptor interaction. So estrogens are involved in the growth, development, and hemostasis of a number of tissues. The physiological effects of these steroids are mediated by a ligand-induced nuclear transcription factor, the estrogen receptor 2. Hormone binding to the ligand binding domain of the ER initiates a series of molecular events, culminating in the activation or repression of target genes. Now, this is a simple where like we can see that there is a ligand or there is a hormone, for example, estrogen, which can bind to its receptor and can form the target genes, and these target genes can then form the specific proteins needed for metabolism. Now, one important is the lock and key model for ligand-gated receptor or for ligand receptor interaction. So, a ligand such as a hormone or neurotransmitter, which is called the key, bind to the specific receptor, which is called the lock, and this binding unlocks the cell's response. Okay. Now, many drugs work by mimicking a naturally occurring hormone or neurotransmitter. If the drug causes the receptor to respond in the same way as the naturally occurring substance, then the drug is referred to as an agonist. So, these are drugs that can pick the lock. Whereas other drugs work in an opposite way. For example, it's an antagonist, and these drugs bind to the receptor, but they do not produce a response. Now, talking about the peak effects as function of dose, you can see that whenever we administer a drug, then with the increasing amount of time, the drug amount in the plasma, the dose in the plasma increases, then it again starts falling as there is metabolism of the drug. Now, talking about dose-response relationship, the weight of an individual is important. Dose is the amount of drug per body weight, which is calculated in milligram per kg. It helps achieve equal blood concentration and often not tightly controlled in humans. So now, talking about dose-response relationship, drug effects are variable. There are group dose-response curve, and they are different for each individual effect because of the temperature, respiration, euphoria, or something like that. Dose response curve. So how response changes as function of the dose. There are accept curves, and the slope is very, very important. The gradual dose response curve is when we increase the dose, there is a small response change, and the steep response curve is when we increase the dose, there is a large responsive change. So we can see here dose response curve in percentage population. So the ED50 or effective dose in 50% of the population is when we administer a dose to the patient, and at least at this point, at least at this dose, the drug is effective for at least 50% of the population administered during the clinical trial. That is called as effective dose. And the lethal dose is when the dose is lethal in 50% of population. We can see in this figure that when we administer a dose, we go on increasing the dose, we go on increasing the dose, and at this point, at this drug dose, it is effective in at least 50% of the population. And at this point, which is a higher dose, it is effective in 100% of population. Now, talking about potency versus efficacy, potency is a relative term, and drug A has same effect as drug B, but at a lower dose. That is called as potency. Now, potency means that, for example, I am administering a drug A, which has the same effect as drug B, but at a lower dose, which means that drug A is potent. So, for example, drug B, we need to administer 500 mg for the same effect, but drug A, we need to administer only 300 mg for the same effect, which means that drug A is more potent. Talking about maximum efficacy, the maximum response magnitude, which means that more drug, no effect if increased. Now, which means that, for example, we are administering a drug, okay, at 500 mg, there is maximum efficacy, and we administer the patient at 600 mg, then the efficacy is not increased, which means that the maximum efficacy of drug is at 500 mg, and there is no point in increasing the dose of the drug, but it might increase the side effects. Now, talking about maximum efficacy, here you can see B has a greater maximum efficacy than A, and A is more potent than B. Talking about therapeutic index, which is the margin of safety. Remember, we are talking about the lethal dose and effective dose. Yes, if we divide the lethal dose in 50% of population by effective dose in 50% of population, then that is called as the therapeutic index. Thank you for watching. Keep studying hard.